guess in this session we first introduced the topic in focus for EHA in the next 4 years, which is precision hematology, and explained the actions that we intended to take to support research, to lobby and network among the key stakeholders to accelerate new formats for clinical trial, to enable multi-arm trials with more precision assays for these patients. And there were several presentations to describe new technologies to detect characteristics in this disease, to classify the disease better, and to decide what type of treatment should be favored. So there were discussions about proteomics to detect the steady state of signaling in cells, and based on this then to make the right decisions. There was discussion of new ways to analyze data, to understand which drug combinations may work best. And then we had presentations about the type of clinical trials that should be fostered in the upcoming years. And the consensus really was that we should come up with a pan-European study based on a master protocol, including functional testing, to explore these new functional diagnostic approaches in the real clinical setting.
Research has significantly contributed to understanding drug resistance in childhood ALL. Can you explain some of the key findings and clinical value from your selected study at EHA?
So we have set up an entire system to explore ALL, so acute lymphoblastic leukemia, functionally working directly with patient material. And I have showed how we can integrate now a large number of really capture the oncogenic program in these cells, and then accordingly to screen functionally for the dependencies on which the leukemias depend most. And I've shown two examples, two subtypes of leukemia at very high risk, already in first presentation, but in particular in relapse, with two examples of possible interventions with new agents targeting in particular the metabolism based on such functional research. Then I've explained our drug response profiling platform, so a platform in which we can at high throughput test the response to drugs to a larger number of drugs directly on patient samples. And with this we can actually get profiles, characteristic profiles for subtypes of leukemia, but also profiles that distinguish more resistant or more sensitive patients. And I've shown several examples how these have implication for clinical management. We have, for example, bridged patients to CAR-T therapy, patients with resistant disease with much less toxicity, so that they come to the CAR T in better shape. And I've shown also approaches to bridge patients with refractory TLL, which is currently really one of the unmet needs, or to CD7 CAR T based on this kind of information.